This is a very complex composition, and it is not totally understood currently. But the main components are proteins for 55%, about 55%, carbohydrates, and so it has been shown that carbohydrates from the cell wall and from the matrix are sometimes distinct. And other components, of course, and finally important, some nucleic acids. And these nucleic acids are called extracellular nucleic acids. And this DNA, we speak about eDNA, and this eDNA is curiously made of mainly non-coding sequences. And this eDNA would be responsible for some exchange of genetic materials between cells that induce the formation of mutants. Finally, regarding Candida biofilms, I spoke a lot about Candida albicans because it is the main species, but other species of Candida are able to form biofilms too, and of course Candida tropicalis, Candida parasilosis, and Candida glabrata, which are also very a lot involved in the mycosis disease, mycosis disease. Just very briefly, Cryptococcus neoformans, which is also a yeast, is able to form biofilms in a special case. In the case of when patients suffer from cryptococcal meningoencephalitis, medical doctors use shunts to manage intracranial hypertension, and these shunts can represent a substrate for the formation of a biofilm. So in this case, we can have a biofilm forming in these patients. So these types of biofilms are not very studied, but they are studied by some colleagues from, especially from Germany, and they have shown, they have published some beautiful pictures of Cryptococcus neoformans biofilms obtained in vitro like that. These colleagues are also described the composition of the matrix of Cryptococcus neoformans biofilm, and it is rather similar to that of Candida albicans biofilm. There are a lot of polysaccharides too. So let's move to the second types of biofilm when there is no substrate. Okay, so in this case we have a host material embedded case. So I will take Aspergillus biofilm as example of this type of biofilm. So in this case, the host material is the mucus, respiratory tract mucus, and this is in the clinical context. This is the case in the clinical context of cystic fibrosis patients, especially. So in this case, there are three main players, key players. The mucus, host component, some bacteria, for example, Pseudomonas aeruginosa bacteria, and fungi, Aspergillus fumigatus, or Cephalosporum, or another fungal species. You can see here some beautiful pictures of Aspergillus biofilms. single-species biofilms obtained in vitro. These features have been made by my colleague Anne Bouvet, who studies Aspergillus at Pasteur Institute in Paris. So she's doing beautiful features. So, in fact, the mechanism of leading to the formation of the biofilm is very similar to that described for Candida. We have the same several steps. First, adhesion, then colonization, adherence, then maturation, and finally dispersion. So the same steps, but here we have some conidia instead of blastospores or organelles. You can see here a biofilm of Aspergillus, showing here the matrix. So you can see here, here again, that this biofilm is an heterogeneous structure. So I will not read all these slides, but just to tell you that the composition is quite similar to those of Candida. The matrix is made of proteins, of carbohydrates, of eDNA, and other components. And eDNA is certainly responsible for the cohesive interactions between cells in the biofilm. Here again, the problem is the resistance of microbial fungi in biofilms, and we have also persisters in this case. So finally, I would like to give you two examples of very different conditions, of example of mixed biofilms. So I mean biofilms including, in this case, two or more species of microbes, and my examples are based on two species biofilms. So there are a lot, numerous different places in our body where fungal species can interact with bacterial species. A lot of different places in the body. Of course, the oral site with oral cavity, respiratory site, ventilator, lungs, the skin, reproductive tract, etc., and so and so. So a lot of different places. And depending on the places, the biofilm will be will include different species. They are not always different species, the same species. The point is that it has been shown very recently that when some patients were co-colonized by two species, the mortality was higher compared with patients colonized by only one microbial species on this place of the body. So this you can see here picture of the team of colleagues. So you can see the percentage survival of the host up to 100%. In red, when the colonization is by only bacteria, in blue only fungal species, and in black, so there is a drastic decrease in the mortality when we have a mixed infection. So biofilms are certainly involved in this increased mortality. When we have mixed biofilms, fungal and bacterial cells have to interact. And these interactions are based on different types of links. For example, physical interactions are already show you it. So physical interaction, direct interaction, but also chemical exchanges. Chemical exchanges means current sensing exchanges. You know current sensing? Current sensing is signaling pathway molecules produced by one species, and these signal molecules are able to modulate another species. So it's an indirect link. We have also interactions based on metabolic byproducts, and so there are different conditions. So altogether, interactions are possible by a lot of different ways in all these microbial cells. So my first example, Pseudomonas aeruginosa plus Candida albicans can interact to form mixed biofilms. But you will see that it is this biofilm is especially good for Pseudomonas aeruginosa and not for Candida albicans, because Pseudomonas aeruginosa kills Candida albicans cells and use these yeast cells as the substrate, okay, as a nutriment, as a substrate to develop its own biofilm. It's a little black, but this is a hyphae of Candida, and you have some bacterial cells, and you can see that they colonize the hyphae, and then they will kill this hyphae and use it as nutriments and as a surface to produce the biofilm. This is a complex slide, but it summarizes the different interactions that I suggested previously. So physical interaction, but also here current sensing, and so on and so. So you can see that here Pseudomonas aeruginosa adhere to the hyphae, kill the hyphae, and develop this biofilm on the hyphae. The killing is shown by the change in color. And my last example is about mixed biofilm based on Staph aureus and Candida albicans. And this is, I think, very important for the future for understanding the physiopathology of infectious diseases. So there are different teams worldwide that study this complex biofilm, including Staph aureus and Candida albicans. We do not study this model, but some colleagues in the United States, in Netherlands, in UK, etc., study a lot it. And very recently, it has been shown that Candida albicans and Staph aureus could form biofilms together, and that Staph aureus did not kill the fungal cells, but used fungal cells as a taxi, a sort of taxi to go inside, to cross the vascular way. Okay? So, in fact, the postulate is that there is a potential synergy in virulence when patients are co-colonized by Staph aureus and Candida albicans in the same place. So, in fact, it has been shown that the bacteria Staph aureus were able to specifically adhere to the hyphae as previously for Pseudomonas aeruginosa. But in this case, they do not kill the cells, the fungal cells. They use them. So, if you look at this picture, it shows the virulence mechanism of yeast. So they cross the epithelial wall and up to the vascular, the blood vessel. And in fact, you can imagine that bacterial cells are adhering to the hyphal part, and so thanks to the fungi, they reach the blood vessels. So the infection is more quick than when there is only bacteria. So Staphylococcus aureus has been shown to invite host tissue and disseminate by adhering to Candida albicans cells. This has been shown both in vitro and ex vivo by the colleagues. And so Candida albicans can facilitate the invasion across mucosal barriers, leading to systemic infection in co-colonized patients. So, and this is my conclusion. The understanding of biofilm mechanism, biofilm development will help us in the future to have a better understanding of the physiopathology of infectious diseases. And once we have this better understanding, it will be more easier. It will be easier to find new therapeutic practices to fight those infectious diseases. Thank you very much for your attention. In fact, I just would like to conclude by showing you our group in Europe. This is called ESGB. ESGB is the HESMID. HESMID is European Society for Clinical Microbiology and Infectious Disease. So this is the study group of HESMID on biofilms, and we have different members from all over the world, not only European people. So if you are interested to be member, it's possible to become. And this is my research team. My research team here with the head of the group here. And this is some pictures showing the pharmaceutical study of Poitiers, my university. Thank you very much.
Thank you, Professor Chrition, for very interesting and inspiring lecture. I believe that this lecture will give you some new knowledge about the biofilms, and a lot of questions you will have a lot of questions about this topic, but we will keep the questions later after the second presentation. The second presentation will be given by Dr. Florence, that will talk about the chemical oxidation by chlorine and in the chemical and process aspect. The floor is here.
Hello, I'm very pleased to be with you this morning. And I'm going to speak about chemical oxidation for water treatment, and especially for drinking water treatment, and especially about two chemical oxidants, chlorine and monochlorine. So to introduce the subject, you know that we need water for a lot of applications. For example, for the energy sector, for the cooling water system, for industrial purposes, and of course for drinking water. Some of these uses need treatment, and often disinfection, oxidation by chemical oxidant, is a key step in the water treatment. So, for these different applications, we need water, and there are two types of resources: groundwater and surface water. And in France, especially, we use a lot of surface water for the energy sector, for the water cooling water system. While for drinking water, we use mainly groundwater because the quality of this water is better than surface water. So, what is the composition? So, what is the composition of groundwater? There is natural inorganic elements coming from the rock dissolution, that is to say, for example, calcium, magnesium, sulfate, chloride. Some of these compounds are indesirable, like iron or manganese, for example, because they give a color to the water, red, for example, for iron and black for manganese. And some of these natural inorganic elements can be toxic, for example, arsenic. I think I have to change because mine is not So if deep aquifers are quite well protected from anthropogenic pollution, we can find in unconfined aquifers anthropogenic pollutants like nitrate, pesticides, organic matter, for example, organic matters, and of course microorganisms. For surface water, there is two types of surface water, rivers, for example, and they are characterized by a low salinity, by non-dissolved compound like suspended solid or turbidity, and by dissolved compound, natural organic matter. Generally, they are highly impacted by pollution, by anthropogenic releases of sewage water, for example. So you can find nitrogen, phosphorus, pesticides, for example, coming from the agricultural practices. And you have also micropollutants, new micropollutants, I will say, endocrine disruptor, pharmaceutical, for example, in this water. Another type of surface water, it is reservoir and dam, for example, and they are characterized by long-time residence. So there is sedimentation from the suspended solid, and you have generally low turbidity and low suspended solid concentration. But other problems like high concentration of natural organic matter and algae or iron or manganese, for example. So to summarize, there is in water suspended solid, turbidity, non-dissolved compound, and dissolved compound like natural organic matter, inorganic compounds, anthropogenic pollutants like pesticides, pharmaceuticals, endocrine disruptor, for example, and also microorganisms, pathogen microorganisms. So to treat water, to produce drinking water from surface resources, we have these different steps. First, pretreatment, then what we call clarification, that is to say these three steps, coagulation, flocculation, sedimentation, and followed by sand filtration. Sometimes we need additional treatment like oxidation, anti-oxidation in these places of the plant, followed by filtration on activated carbon, and then disinfection between the supply of the distribution system. So first, pre-treatment. Pre-treatment consists of screening, that is to say a filtration of the water through a grid, like this. And there is a system to clean mechanically the grid, and the waste are collected in the trash box here. It is necessary to remove largest waste, like branches or like bottles, for example, which can be present in the resource. Then the clarification. The clarification is the three steps: coagulation, flocculation, sedimentation, and sand filtration. Coagulation flocculation is the addition of a chemical based on iron and aluminium to allow the agglomeration of the small particles in larger and heavier flocks, which can precipitate later. So this is a lab experiment with ferric chloride as a coagulant, with ferric chloride as a coagulant, and you can see the agglomeration of the particles